వాయిస్ వినిపించట్లేదు నెట్ కనెక్ట్ అవ్వాలి కంప్యూటర్ మిగిలిన 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 వాయిస్ వినిపించట్లేదు నెట్ కనెక్ట్ అవ్వాలి కంప్యూటర్ మిగిలిన
స్టార్ట్ ఇచ్చారు వీళ్ళు.
Pawan, how are you?
Sir, I am good, sir. How are you, sir?
Right, fine.
Yes, sir. Very nice to see you after so many days, sir.
After long gap.
Yes, yes, sir. Yes, sir.
Yeah, okay. Monica, start channel.
Sir, shall okay, sir.
Yeah, start channel.
Good afternoon, respected participants and distinguished guests. I'm Monica, your today's host. I'm delighted to welcome back you all to day 3 of a one-week online National Faculty Development Program hosted by Sir C.R. Reddy College of Pharmaceutical Sciences. It's a pleasure to have all of you here today, and we are excited to engage in some enriching discussions and learning experiences. We have had excellent presentations on the first and second days by Dr. Hanmi Reddy, Director at Slayback Pharma, Hyderabad, and Dr. Sheikh Afzal Basha sir, working at St. Mary's Group of Institutions, Guntur. As we gather here for this intellectual endeavor, I would like to take a moment to congratulate our Indian cricket team for securing a remarkable victory yesterday. Let this achievement serve as a testament to the Indian spirit of excellence and dedication that we all strive for. Let's instantly channel the same spirit into our FDP as we continue with our today's session. We have a fantastic speaker lined up, and there's much to be gained from his expertise. Thank you all for being a part of this exciting journey of learning and growth. Today we are honored to have with us a distinguished guest, Dr. S. Pavan Kumar, Associate Professor at Sri Vishnu College of Pharmaceutical Sciences. He has a wealth of knowledge and experience in the field of pharmaceutical sciences, and he will be sharing his treasured insights on our topic, empowering faculty excellence through enhancing research, competence, and innovation. I would like to invite Ms. Yamuna to introduce our today's speaker to the gathering.
యమున అన్మ్యూట్ చేయండి. అన్మ్యూట్.
Thank you, Yamuna. Without any more delay, I would like to hand over our session to the most awaited Dr. S. Pawan Kumar, sir. Sir, please take over this.
Thank you so much. Yeah. Very good afternoon to all the participants and distinguished guests. Yeah, first of all, sir, I would like to express my sincere thanks to Principal Sir Narendra Sir. He is my guru for providing this wonderful opportunity to present my research experiences in this particular podium. So I'm very thankful to sir. So and thank you, Yamuna, for nice introduction. So we're going to session. Yeah, is it visible?
Yes, sir. Yes, sir.
Yeah. Okay, thank you. Yeah, very good afternoon to all. So welcome to the third day of the session. So today I would like to share few of my research experiences. So first of all, this is really very interesting FTP. So empowering faculty excellence, enhancing research competence and innovation. Yeah, it's absolutely true that nowadays we will get excellence in the field of academic profession with only research competence and innovation only. So without research and without innovation, we cannot excel in our field. So really it is a wonderful topic. So what I can say, if you see any kind of academic performance score or academic performance index or anything, research is very important. So so everyone has to focus on the research in their levels. So it's a very good topic and I'm very enlightened to share this information. So today I would like to share a few things about my research, that is novel role of PPARs in the brain. So they're promising targets in the therapy of Alzheimer's disease. So first of all, coming to the, so just 1 minute. So why what is the objective of my research? Then next, in case of the purpose of the research means so obviously so many of us we are doing our research academic level, we are doing our research. So in case of the academics, why we are doing research means one of the main thing is in the research, there are various steps used to be there. Okay, so our ultimate aim is to establish good lab. Okay, to establish the good laboratory, we need to have the good funds. So when to get the good funds, we need to have the good papers. Okay, so this is all is like a cyclic process. So if we will have a good number of papers, quality papers, then only we can able to apply the projects. Means of course without having papers also we can able to apply the projects, but the thing is we will have a papers, then only our projects get approved. If we can able to get our projects, then only we can get the funds. With the funds we can able to do the good research. So first of all, we need to know the purpose of the research. So our main goal is to publish the papers. One of the main goal in the research is it is two ways. One is maybe for the patent, another one maybe for the publications. So this is the why we are doing research. Okay, of course this is the at the level of the faculty, these are our goals, and at the same time at the level of the students, what students what achieved from this particular research means they can get the good knowledge about the research. They know how to design the projects, and also we are providing good manpower to the industry. So these are all of the different goals of the research because of the Yeah. Is it audible?
Yes, audible, sir.
Yeah. So then one more thing is like selection of the drug and its rationality. So which kind of drug we are selecting. So selection of the drug plays very important role in our research. So previously we used to select if any drug is having antioxidant activity, so then based on that we can do like all type of anti-diabetic or anti-cancer or all the studies. It's okay, we can do. But nowadays, like if you want to publish our paper in good journal, then it should have some a molecular level connection also. So always there should be some rationality when we are selecting the drug. Then experimental design. So experimental design, it plays very important. So if you want to publish your study in any good journal, then definitely you should have good amount of the data. Okay, so in case of the pharmacology or in any areas, so we need to have the good amount of the data. If your study has to publish in the good paper, good paper means indexed paper, or in case of the impact factor paper. So always better to go for the impact factor paper. So in case of the experimental design, in case of the pharmacology point of view, in any study, if you have behavioral studies or next biochemical studies, then molecular type of studies, then histopathology or immunohistochemistry. So those kind of studies plays very important role. So if you have all those kind of data, then it gives a lot of weightage to the paper. Then after completion of our study, then next and foremost important thing is the analysis of the result. So whatever the data so far we got, so we have to analyze that particular result. Okay, so for analysis of the result, there are n number of softwares are available. Among that, one of the thing is the Graph Prism or SPSS also you can use, or simply in Excel also we can perform. Then so many what is the what is our what I can say. So because of our so many commitments, so maybe we can able to achieve till analysis of the result, and also maybe we can able to complete the what I can say thesis submission, all those kind of things also. But so much what I can say so much of us we are leaving our study in that particular point itself. So very few people they are going to the next level to write the manuscript. So please try to remember one point: without publishing our study, there is no value for our study. Okay, so whenever we will publish, then only our study gets some recognition. Okay, so whatever the journal, okay, at least try for the good impact factor. If not possible, at least try to go for the index journal. So that is sufficient. But don't keep all your study on your bench. Okay, so please try to write the manuscript and go for the publication of the paper. So so in any research, okay, these are the steps. So first of all, we need to design our study, we have to execute the study, then we have to go for the analysis of the result, then need to write the manuscript and go for the publication. So please try to remember, it is very toughest job, but there is no shortcuts. Okay, always better and always it is advisable to publish our study, whether it is in indexed journal or impact factor journal. It's okay. So first of all, we need to publish our study. Okay. So with this introduction, so like I will try to explain one by one. Okay. So here, like we have selected one of the disease, that is the Alzheimer's disease, for my research. Okay. So why I have selected Alzheimer's disease means, already as I point out that, so like we need to select based on the availability of the resources. So we have very good, like what I can say, neurobehavioral lab. So it is meeting my criteria. And one more thing is that Alzheimer's disease. So as per the statistics, what statistics are suggesting that it is one of the, like maybe like as per the CDC or World Health Organization. So both the agencies, they declared that Alzheimer's disease, it is one of the most devastating neurodegenerative disorder in older adults. So especially like we have the data till 2007. As per the 2017 WHO report, approximately 5 crores people, approximately 5 crores people are suffering with the Alzheimer's worldwide. So this particular, maybe it will be more time progresses. So maybe like the data is suggesting that it may reach 150 millions or more in case of the 2050. Okay, so coming to the, like, as everyone knows, I'm not going to give much importance to the introduction of the Alzheimer's disease. So Alzheimer's disease, it is a irreversible progressive brain disorder. So main thing is here, like dementia is one of the, like initial symptom is the dementia, later it will cause so many things. So like this is the, like the progression of the Alzheimer's disease. So maybe it may take 10 to 12 years to reach the severe stage of the Alzheimer's disease. So whatever the treatment modalities we are having, so they will work when we will identify at early stage, so that at least we can what I can say, we can attenuate progression of the disease. Okay, so always, so this is the thing. So but no drugs, currently no drugs are available to treat the Alzheimer's at the later stages. So yes, with the available drugs we can able to treat at the level of the mild Alzheimer's. Here we cannot able to reversible the disease. At least what we can do, we can inhibit the progression of the disease. So initially, like people may suffer with the short-term memory loss, that is the initial symptoms. Later, problem with the reading, and also they couldn't able to identify the objects, poor object recognition, then poor direction sense. Even they couldn't able to reach the, like very well-known places also in how we will reach. So poor direction senses. Like when it will reach to the moderate kind of stage, then their judgment also used to be a very poor judgment. And also attention, they couldn't able to concentrate on anything. Of course, nowadays, like one more term has come, like scientists are calling one more term, that is the digital amnesia. So digital amnesia also same thing, like what I can say, like because of the usage of the gadgets also, previously Alzheimer's used to get after 60 years or 65 years, like that. But because of the usage of the gadgets, people are prone to this kind of the cognitive impairment related ailments, maybe at the early ages also. Then in case of the severe case, so even people will suffer with the visual problems. So why like all these things are happening in case of the Alzheimer's disease? It is because of the, so if you see these things like, see the normal neuron. So in case of the Alzheimer's disease, neuron used to be visible like this. Okay, so why it is happening means because of the formation of the amyloid beta plaques and neurofibrillary tangles. These are the phosphorylated tau proteins. So these are the main villains in case of the Alzheimer's disease. One is the beta amyloid plaques and another one is the tau proteins. So whatever the research nowadays it is going on, mainly all the scientists they are targeting the beta amyloid plaques. So because of that, like recently FDA got approved one drug, monoclonal antibody. It will act on the beta amyloid plaques. That is the aducanumab, and the brand name is the Edihilam. So they have released that particular drug into the market, and they are thinking that it may do wonders in case of the Alzheimer's disease. So it will directly on the amyloid beta plaques, and it will cleave the amyloid beta plaques. So in case of the Alzheimer's, main targets are the development for the development of the Alzheimer's, two things are there. One is the beta amyloid plaques, and one other more is the neurofibrillary tangles. So then further, like so many studies, what they are suggesting that if we will see the brains after postmortem, during the postmortem. So postmortem studies on the human brain, they are suggesting that So the main role for the development of the Alzheimer's is because of the formation of the extracellular amyloid plaques and neurofibrillatory tangles, oxidative stress, then mitochondrial dysfunctions, then inflammatory reactions, and apoptosis. These are the main reasons for the progression of the Alzheimer's disease. Okay, sorry to bother you with the initial introduction, but without this introduction, we cannot proceed. So that's why just given small recap regarding the Alzheimer's disease. So like if you see this is the thing. So like amyloid plaques in the formation of the amyloid plaques in the neurons, then formation of the neurofibrillatory tangles within the neurons. So this for formation of the beta amyloid plaques. So why beta amyloid plaques are forming means, so like it may be because of the amyloid precursor mutations in the amyloid precursor protein, or it may be because of the mutations in the PSN genes, or it may be because of the mutations in the apolipoprotein E4. So all these three, they may cause the formation of the beta amyloid plaques. So here, like one of the main, like out of these three, also one of the main important one is the amyloid precursor proteins. Okay, so these amyloid precursor proteins are very, very important. So like amyloid precursor proteins, I will explain later about this metabolism of the amyloid precursor proteins also. This amyloid precursor protein, it leads to the generation of the beta amyloid plaques. Then this beta amyloid plaques may causes dysfunction of the mitochondria or development of the iNOS. So this is like inflammatory mediators like generation of the free radicals, oxide free radicals, oxygen free radicals, and so this beta amyloid plaques. So it may as I told you, these are the main villains in Alzheimer's disease. This beta amyloid plaques, in one way, what they will do, they will generate the free radicals, oxygen free radicals as well as the nitrogen free radicals. So that here formation of the oxides, oxygen free radicals as well as the nitrogen free radicals, and they lead to the development of the oxidative stress. Further, this beta amyloid plaques also causes neuroinflammation. So this neuroinflammation means beta amyloid plaques, they will activate the nuclear factor kappa beta, NF kappa beta. This is one of the main transcription factor in inflammation. So this NF kappa beta, beta amyloid plaques cause activation of the NF kappa beta. This NF kappa beta also further lead to the release of the N number of inflammatory mediators like TNF alpha, interleukin 1 beta, all these kind of the inflammatory mediators. And ultimately it lead to the cytokine storm, or otherwise we can also call as the neuroinflammation. So all together they causes the oxidative stress. This oxidative stress lead to the apoptosis. As we all are aware that apoptosis means programmed cell death. So it lead to the apoptosis. So neuronal cell death used to happen. So neuronal cell death means it is neurodegeneration. Neurodegeneration lead to the development of the Alzheimer's disease. So here main culprits are the beta amyloid plaques. So first of all, whenever we are doing any research, we need to concentrate on the pathophysiology of the disease. So then only we can able to identify good targets to mitigate that particular disease. So in case of the Alzheimer's disease, so if whatever the drugs, whatever the agents, so how it should when it will get benefit in case of the Alzheimer's disease means whatever the drug we will select, if it will act on the beta amyloid plaques, yes, then we can stop all these downstream processes, then we can get some effect, some beneficial effect in case of the Alzheimer's disease. Or any agent, if it can able to act on oxidative stress, or if any agent, it can able to act on the inflammation, or if any agent, it can able to act on the tau protein phosphorylation, then we can get some neuroprotection and we can stop the progression of the Alzheimer's disease. So all the scientists throughout the world, they are searching for the agents which will act on the amyloid plaques. Okay, so in this connection, so already I have explained, so these are the different factors, they will cause the Alzheimer's disease. One of the main factors is the amyloid plaques and the phosphorylation of the tau proteins. Then these two, they cause the mitochondrial dysfunction, neuroinflammation, oxidative stress, all together they lead to the Alzheimer's disease. And of course, all this neurodegeneration, especially it is happening in the cholinergic neurons. Neurogeneration, it is happening for the cholinergic neurons. So in case of the Alzheimer's disease, diagnostically, three things used to happen. So if you see the levels of the neurotransmitters, the levels of acetylcholine used to be drastically reduced. The levels of the acetylcholine, decrease of acetylcholine levels, then increase of the beta amyloid plaques, and increase in levels of the phosphorylated tau proteins. So these are three are the main factors. So in this connection, there is different strategies we need to develop to mitigate the Alzheimer's disease. So whatever the drugs, okay, so that can able to act on the beta amyloid plaques or oxidative stress or neuroinflammation, they will be good target to treat the Alzheimer's disease. So in this prospect, when we are searching the literature, we found an interesting thing is that there is a huge relationship between the PPR alpha, as we are aware that the PPR, so peroxisome proliferated activated receptors. So PPRs, they are very much linked with the Alzheimer's disease. So when we have done the literature, we found that in case of the Alzheimer's disease, like when we have done gene expression for in case of the Alzheimer's disease, scientists found that the expression of the PPR alpha and PPR gamma got significantly reduced in case of the Alzheimer's disease. Then, so at the same time, PPR alpha as well as the PPR gamma, they are also effective against the amyloid beta proteins also. So this is the general thing, like role of PPR alphas in pathophysiology. So like already PPR alpha agonists are available in the market. We are using fibrates for the hyperlipidemics. Then PPR gamma agonists are also available in the market. Those are like glitazones, 5-glitazone, rosiglitazone. We are using for the anti-diabetic. But there are new strategies are available. New research is going on to develop PPR alpha as well as PPR gamma agonist to treat the Alzheimer's disease. So this is the thing. So PPR alpha, so what it will do means PPR alpha, it will regulate the metabolism of the amyloid precursor protein. As we have already studied that amyloid precursor protein. So this amyloid precursor protein, it plays very important role. Amyloid precursor protein plays very important role in the development of the Alzheimer's disease. So this metabolism of the amyloid precursor protein, this amyloid precursor protein used to be cleaved by the two enzymes, two protease enzymes. That is, one of the enzyme is the alpha secretase, another one is the beta secretase enzyme. One is alpha secretase, another one is the beta secretase enzyme. So here what is happening means, if alpha secretase can able to cleave the amyloid precursor protein, then it lead to the non-amyloidogenic pathway. So whatever the metabolites will be formed when it will be follow the alpha secretase pathway, those metabolites are neuroprotective. They are very beneficial in nature. Instead of that, when this amyloid precursor protein is cleaved by the beta secretases, whenever this amyloid precursor protein are cleaved by the beta secretases, then it lead to the amyloidogenic pathway and it lead to the generation of the amyloid beta proteins. So this is the interest. These are the findings we found in the literature. So any agent, it can able to activate the alpha secretases, or any agent, it can able to downregulate the beta secretases. So if we can able to target these things, then we can able to achieve good medication to treat the Alzheimer's disease. So interestingly, PPR alpha, okay, the agents, the drugs which can able to stimulate the PPR alpha receptors, they can able to upregulate the expression of the alpha secretases. So PPR alpha agonist, what they can do, they can able to upregulate the alpha secretases. So if any drug can able to upregulate the alpha secretases, then it lead to the non-amyloidogenic pathway. So we can able to attenuate the Alzheimer's disease. At the same time, these PPR alpha agonists, they can also able to downregulate the beta secretases. So because of the downregulation of the beta secretases, we can also prevent the generation of the amyloid beta plaques. So if we can able to inhibit the amyloid beta plaques, then there is no neurotoxicity, no neuroinflammation, no neurodegeneration, and we can stop the progression of the disease. So in that way, this PPR alpha plays very important role to attenuate the Alzheimer's disease. So this is the literature. It is suggesting that in case of the Alzheimer's disease, the levels of the PPR alpha is downregulated. The levels of the base, as we have discussed, beta secretases, it is also called as the base. The levels of the base is upregulated. So because of these things, what is happening? Because of the upregulation of the base, it is leading to the amyloidogenic pathway, and accumulation of the amyloid beta plaques are increasing. At the same time, because of the decreased levels of the PPR alpha, it is causing the decreased expression of the alpha secretases. Further, it is leading to the amyloid precursor protein. So if any agent is able to stimulate the PPR alpha, then we can reverse all these things and we can control the progression of the Alzheimer's disease. So these are from the literature, we found that these are the things PPR alpha can able to cause all these things. PPR alpha can able to inhibit the oxidative stress. PPR alpha can able to attenuate neuroinflammation. PPR alpha can able to attenuate the amyloidogenic pathway. At the same time, PPR alpha can also attenuate the hyperphosphorylation of the tau proteins. So based on this, we found that if any agent can, if any drug, if by using any agent or any drug, if you can able to activate PPR alpha receptors, then these are the good targets to treat the Alzheimer's disease. So this is the what I can say. This is one of the strategy for selection of the drug in our research. So this is the thing, one of the rather than simply based on the oxidative stress, we have to dig out the literature and we have to know what are the possible pathways are involved in that particular disease. Then based on the pathways, if any pathway, if any agent, if it can able to moderate that particular pathway, we have to select those kind of drugs for our research. Then it will add a lot of rationality for selection of the drug. So yeah, is it audible?
Yes, sir. Audible, sir.
Okay, thank you so much. So almost so how to select the disease, then how to select the drug. So I hope it is not that much complicated, but this is the way to select the drug for our research. Okay, so based on the pathophysiology, based on the pathways, we have to select our drug. Whatever the drug we are selecting, it should have some link with the pathophysiology of our disease. Okay, so now I think you can able to understand that. So what are the agents we have selected? What are the drugs we have selected for our research? So we have selected the drugs which can able to activate the PPR alpha receptors because they can able to attenuate neuroinflammation and also they can able to downregulate the beta secretase agents. So because of that, we have selected the PPR alpha agonist. And one more thing is there is also involvement of the PPR gamma in Alzheimer's disease. It is not only limited to the PPR alpha. Surprisingly, PPR gamma also playing very, very important role in the Alzheimer's disease. So PPR gamma, it is mainly PPR gamma where it is playing important role means. So so already this entire story already have discussed. So because of the formation of the amyloid plaques, what is also happening means these amyloid beta plaques, they are causing the neuroinflammation. So because of the neuroinflammation, neuronal cell death is happening. So our PPR PPR gamma agonist. So PPR gamma agonist, they are very good to control the neuroinflammation. So if you will select PPR gamma, by using PPR gamma, we can control the neuroinflammation. By using PPR alpha, we can control the base expression. So in this point of view, what we have selected is the drug which can able to act on both PPR alpha as well as the PPR gamma. Means we have selected the dual agonist, PPR alpha as well as the PPR gamma agonist. Based on this, we have done a lot of literature search also. So far, no one has done these kind of studies. The drugs which are stimulating the PPR alpha and PPR gamma, no one is explored these kind of studies in Alzheimer's disease. So we have selected this kind of drug. And also PPR gamma. So this is PPR gamma activator, PGC1 alpha. This is the activator of the PPR gamma coactivator. So PPR gamma coactivator, what it is doing, it is also activating the NRF2. NRF2 is one of the important antioxidant transcription factor involving in the antioxidant mechanism. And also PGC1 alpha, PPR gamma, it is also activating the BDNF. BDNF means brain-derived neurotrophic factor. This is responsible for the neuronal growth. It will, if any agent can able to increase the levels of the BDNF, then it will increase the health of the neuron. So it will show good neuroprotection activity. So in this point of view, we have selected PPR. Hence, the drugs which can activate both PPR alpha and PPR gamma receptors can be investigated for use in Alzheimer's disease. So in this point of view, we have selected the drug called as the saroglitazole. Saroglitazole. So this saroglitazole is the STZ. It is a dual agonist. It can act on the PPR alpha as well as the PPR gamma. It is FDA approved drug. It is approved for the diabetic dyslipidemia, but so far no studies were conducted on the Alzheimer's. So hence we would like to investigate the role of the saroglitazole in Alzheimer's disease. Yeah, so far so this is the if anyone have any doubts, please you can interrupt me and you can ask me. So in this way we need to select the drugs for the to target the diseases. Okay, so selection of the disease plays very important role. Then selection of the drug, this is the heart of the study. So based on the drug, whatever the drug we are selecting, it only plays important role in the research. Okay, then coming to the experimental design. Okay, so whenever we are designing the experiment, so now my test drug is I have selected my test drug and I have selected already the disease also. Now I would like to explore the activity of saroglitazole or the Alzheimer's disease. So for that, what I have to do means I have to develop first one animal model. I have to develop. Okay, so I have to develop the animal model. So whenever we are designing the experiment, okay, so please keep in the mind that so whether our test drug may get activity or not, that is different story. Okay, so maybe sometimes we can succeed with our hypothesis, sometimes we may fail. Okay, that is not the big deal, because if one drug will fail, then we can select the another drug and we will continue our research. But one of the heart of the research is that cell development of the model. So at least we have to we can able to develop the model. So for that, we have to standardize the model. So for standardization of this study, now, for example, any researcher, now maybe now you are thinking that, yes, I want to do any study on the Alzheimer's disease, or otherwise I want to do my research on the diabetes, or I want to do my study on the cancer. So first, what is the main thing is you have to develop the model. You have to develop the model. Development of the model plays very important role. If you couldn't able to develop the model, then there is no value for your study. So for that, what we need to do means, first of all, we have to conduct pilot studies. So for example, if it is already established model, then it's fine. If it is not established model, we need to establish that particular model in the laboratory. So for that, again, we need to dig the literature. Maybe for example, I will share my personal experience in this particular study. So if you see the literature, so literature is suggesting that here for this study, I have selected scopolamine model. Okay, by using scopolamine, there are different models are there to induce the Alzheimer's. One is scopolamine induced Alzheimer's, arsenic induced models are there, or stress induced models are there, or otherwise diabetes induced cognitive impairment models are there, or what I can say, steroids induced models are there. Different models are available. So for my study, I have selected scopolamine model. Okay, in this scopolamine model also, if we search the literature, so different doses are there, like maybe 1 mg per kg, 2 mg per kg, 3 mg per kg, 5 mg per kg, different models are available. And at the same time, there are also reports that few people have done within 1 week, they have completed the study. Few people within 3 days, they have completed the study. Few people, they have completed 14 days, 19 days, like that they have. So, even with the literature, we can get so much, what I can say, based on their lab and based on their requirement, they used to select their models. So that's why you have to design your model. Okay, you have to design your model. So with all these permutations and combinations, and with the pilot study, we have standardized our model. For our model, we have established our model at the 3 mg per kg. Okay, so for this one, we have done pilot study with 1 mg per kg, 2 mg per kg, 3 mg per kg, 5 mg per kg, with the different doses we have conducted the study, and we achieved with the 3 mg per kg. We have established a very good model for the Alzheimer's with 3 mg per kg. Okay, so this is the pilot study. This is the pilot study what we have conducted. Then already I told you, for any good pharmacological study, there are 4 steps are there. One thing is that data, how we can able to what is the data we need to generate in our study means. First thing is if it is neuro-related study, behavioral data is mandatory. We have to do whatever the behavioral parameters are available in our lab. means like suitable, which are suitable to our study. We have to perform all that particular behavioral test. Okay. So 4 things are very, very important in whenever you are designing any studies. Please try to remember this point. It is very, very important. So data, we need to generate the data if we want to publish our paper. Please don't get bored by I'm using that word like publishing the paper, because like without publishing the paper, we cannot excel in our field. Okay, for everything, publishing the paper is very, very crucial. So that's why I'm saying. So why we are doing all these things means like already I told you to train the manpower, to train our students. That is one thing, and to equip the knowledge and to improve or to enhance our knowledge and to publish the paper. So these are the objects. So for publishing the paper, we need to generate the data. So like for any study, what is the data we have to generate means? So if it is CNS related study, then obviously first and foremost thing is the behavioral data. So we need to develop the behavioral data. Then after behavioral data, then biochemical data. Okay, biochemical, whatever the test we are performing by using the tissue or blood. Okay, biochemical data is very, very important. Then molecular level data. So in case of the molecular level data means we have to find out the expression of the genes. Okay, how genes are expressing or how proteins are expressing. So we need to perform the gene expression studies or protein expression studies. Okay, at the level of the genetic level or at the level of the protein, we have to perform our studies. This is at the level of the molecular. So we have to, so for example, see if in case of the BFARM level studies, so we can stop at the level of the behavioral data. So for example, if it is the thesis of the MFARM, then what we have to do, we have to include both behavioral as well as the biochemical data. No, it is not for the MFARM also. It is if it is for the PhD thesis. For example, if any one of you are enrolled for the PhD, then what we should not stop at the level of the biochemical level. We have to go at the level of the molecular level. So like why molecular level means like entire my presentation, what I can say, I'm rounding around the beta secretaries, base expression, or otherwise also I told you BDNF level. So these are the proteins. So we need to perform the studies to determine the expression of those particular proteins in the sample. Okay, so like we have to perform the molecular level studies also. So for molecular level studies, there are like different techniques are available. Maybe simply like we can perform the ELISA studies by using the plate reader or ELISA reader, like you can find in this particular slide. Or if you want to see the gene expression, then we can go for the RT-PCR, reverse transcriptase polymerase chain reaction. Or if we want to go for the protein expression, then we need to go for the Western blotting studies. Okay, so in that we need to fix like if it is BFARM, then we can restrict to behavioral studies. If it is MFARM, there is no wrong if MFARM students also if they will perform molecular level studies. Okay, so far like what I'm trying to say is like we have to develop the behavioral data, and also we have to generate biochemical data, and also we have to develop the molecular level data. And at the last, and one more important thing is the histological data. Okay, so for this, like maybe we can go for the histopathology or we can go for the immunohistochemistry studies. So whatever the study you are doing, maybe behavioral studies are not possible to include in all the studies, but at least in all the studies we have to or need to include the biochemical data, molecular level data, as well as the histological data. As per our experiment, so here what we have done, we have selected the test drug is the Saraglitazar, inducing drug is the scopolamine. So here we have performed different behavioral tests on different days. So one is the open field test we have performed, novel object recognition test, NORT we have done, then BIMAGE we have performed, then Morris water maze we have performed. So these are the behavioral tests we have done. So after completion of all the behavioral tests, at the last day, on day 16, we have sacrificed the animal and we have collected the tissue and we went for the performance of the biochemical test and the histopathology. So this is our experimental design. Okay, then after experimental design, already I shared the. So then we will generate the lot of data. Okay, we will generate lot of data after conducting all these experiments. Then what we have to do? We have to analyze the things. Okay, we have to analyze our data. We have to analyze our data. Without analysis, no data will get we cannot like there will not be any validity for the data. So there are n number of, I cannot say n number, there are many tools are available to analyze the data. In that, one of the best tool is the GraphPad Prism. Other than GraphPad Prism, there are like already I told you, like SPSS is there, Excel is there. So with the different this tools we can use to analyze our data. So just what I can say, I will share one thing. So for example, like so for example, like so I generated this kind of data. Okay, for example, like here I'm showing the data of the Morris water mist test. So here these are my groups. For example, let us say that control group, scopolamine group, then standard group, that is gonoprazole group, or otherwise like some tests. So these are the different groups. So for example, here we can say that the control, disease, standard, or test. These are the groups. And here, what is my parameter means escape latency. Like how, like later I will explain about those details. So this is my data. So now how to analyze my data? Okay. So otherwise I will share related to these things after come. Yeah. First I will explain about the. So for example, let us say that first we'll go to the. So this is the one of the tests we have performed. This is the Morris water maze test. Okay, so in case of the Morris water maze test, so please see this video once, then you can able to understand. So we will. This is the maze. We will place the animal. So see. Yeah, hope I think you might have seen this video. Like, so video play in the day.
Yes, sir.
Okay, so like in case of Morris water mesh test, what were like what is the parameter? What we're estimating here means how much time, whether it is mice or rat. In our study, rat we have taken. So how much time rat is taking to reach the platform? So this is called as the escape latency. How much time rat is taking to reach the platform? Okay, how much time rat is taking to reach the platform? This is called as the escape latency. So by using like Morris water mesh, we will so see if you see these trackings. So these are the trackings. It will like by using Maze Master software, we are doing this kind of experiments. So all these trackings we got in that particular software. Okay, then so I hope. So this is the Morris water mesh test. So here we will like virtually we will divide this maze into 4 quadrants. In one of the quadrant we will place the platform and we will drop the animal in any one quadrant. Then we will note down the time, how much time it is taking to reach the platform. If any animal, if it is having the good memory, then what will happen? It will not take much time to reach the platform. Actually, like to perform this kind of experiments, we have to train the animals at first. First, we need to train them. So like maybe it may take 2 or 3 days for training. Then after training, we will perform the test. So if any animal, if it is having good memory, then immediately it will not take much time to reach the platform. If animal, if it doesn't have the memory, then it will try to search for the platform. It will take a lot of time to reach the platform. So after administration of the drug, if animal could able to reach the platform at the earliest, then we can say that our drug can able to attenuate the depression of the memory. Okay, so our drug can able to attenuate the Alzheimer's disease. So now, so for this, I will I will explain how to generate the data. So this kind of graphs we need to like this is the analysis of the results. We need to produce this kind of the graphs. Okay. So this is the data we got after completion of this experiment. So maybe in case of the controls, if you see the results, in case of the control animal, first animal it has reached within 15.5 seconds. Second animal also took 15 seconds. Third animal took 16 seconds. Fourth animal took 10 seconds. 10 seconds, 11 seconds respectively. So this is in case of the control animal. But in case of the disease group, if you see the disease group, it took first animal, it took almost 40 seconds. The second animal also almost most of the animals took 40 seconds. Fifth animal took the 47.5 and sixth animal took the 62.5 seconds. So like blindly by seeing this result, we can say that yes, diseased animals took more time to reach the plant. Simply if we say it took more time to reach the platform, it is not sufficient scientifically. Okay, we need to derive the whether this particular difference is statistically significant or not. Okay, so whether this difference is statistically significant or not, that one we have to calculate. Okay, so for example, with our naked eyes and with our limited knowledge of the calculations, we can say that yes, disease group taking more time to reach the platform. So we can say like it is taking more time to reach the platform. Simply if I give statement like that, it is not sufficient. We have to perform the statistics. After performing the statistics, if it is really statistically significance is there, then only we can say that this animal is suffering with the cognitive impairment. Then see the data of the test drugs. So in case of the, for example, like test drug, after administration of the test drug, first animal it took 19 seconds, second animal 32 seconds, next 24 seconds, fourth one is 5 seconds, like that. Like this is the data, what I can say, like this is the data for the test animals. So like if you see like this, like we can say that yes, it is taking maybe comparatively less time to reach the platform when compared to the disease group. So for that, whether there is a significant difference is there or not, for that we have to perform the statistics. So to perform the statistics, we have to go for the different we can use SPSS or we can use the graph prism. So like if you see the graph prism, so it will be like this. So this is the opening page for the graph prism. This is the opening page for the graph prism. So here there are like different sections used to be there, like XY column group, like the different sections used to be there. So for example, like we can also import the data. So like for your convenience, I will do one thing like so like here one more thing I also need to share with you, like generally in our pharmacology related studies, we will use two types of ANOVA test. One is t-test, another one is the ANOVA test. ANOVA means everyone is aware that analysis of variance. So if there are only two groups are there, then we will go for the t-test. More than two groups are there, then we will go for the ANOVA test, analysis of variance. In ANOVA, further one-way ANOVA is there, two-way ANOVA is there. I don't want to go in depth, but simply we can use t-test if only two groups are there. We can use ANOVA if more than two groups are there. In this case, so there are four groups are there: control group, disease group, then standard group, then per se group, test group. Total 5 groups are there. So as 5 groups are there, we need to go for the, as more than 2 groups, we have to go for the ANOVA. So here we need to go for the ANOVA test. So here XY is there, column is there, group is there. So select the either column you can select or group you can select. Then see the things. So if you are having mean as well as the SEM, then you can select this one. Enter and plot error values already calculated elsewhere. Maybe by using Excel, if you have already calculated mean as well as SEM, then directly you can choose this one. So here like subsections are there: mean, standard deviation, N or mean, standard error mean, N means like how many number of animals you have used for the group. Okay, total how many number of animals you have used for the group. Like that we can go. So for example, if you don't have mean as well as the SG, then we have to only, as I have shown in the Excel, I have crude values, raw data I'm having. Then we need to select the enter replicate values in side by side subcolumns. So see, as I'm having 6 animals data, I have to select the 6. Then I have to create the table. Okay, so this is the table I have created. So here, what is the parameter I have selected? Escape latency. Then if I go for the groups, so see, for example, these are the groups control. This is okay. For demonstration point of view, only 4 groups I kept. So these are the 4 groups: control, disease, standard, and test. So control group, disease group, standard group, and these are the 4 data sets. Then simply we need to paste the data sets. So in case of the control group, we need to select these 6 values to paste in the result. Okay. Then in case of the disease also, just select the disease data set and paste. Okay. Then standard, just select the standard data sets and paste. Then in case of the test, select the test data set and paste. Okay. So now, so I'm measuring the escape latency and my groups are control group, disease group, standard, and test. As 4 groups are there, which test I have to do means I have to go for the ANOVA test. So how to perform this one? So here, for example, like for simplification, like in case of data, you can edit and you can write the as this is the Morris Watermesh test. You can type the Morris Watermesh. So next thing is the analysis. So see here, there are different tabs are there. Different options are there. Here, one thing is analyze the data set. So click on the analyze the data set. So then if you click on this, see under the column, there are like a t-test, ANOVA, one-way ANOVA, T and Wilcoxon test, descriptive statistics, like different tests are highlighting. And what is the test we need? T-test or one-way ANOVA? Which test we need? Anyone? For this data, as more than 2 groups are there, ANOVA test. Yeah, so just click on the ANOVA. So ANOVA, we have to click on the ANOVA. Then these are the sets are there. How many sets? 4 sets are there. Control, disease, standard, and test. Then just simply press the OK. Then it will give. So here it is like no pairing. Everything is independent data. So it is already saying that here 2 tests are there. Whether we are going for the ANOVA or it is non-parametric test. So we have to do the ANOVA. Yes, default we can keep yes, use ANOVA. And assume the is equal standard deviations. Yes. So this is the thing. Then after that, repeated measures. We have to go for the repeated measures. So nothing like here no need to select anything. Then this is the one more most important thing, and we need to get confused here. Multiple comparison test. So multiple comparison test means like we need to compare with which group with what. So like in any pharmacology data, so like what is our objectives? As I told you, first thing is we need to develop the disease model. So first of all, what we need to check whether disease got induced or not. Okay. So for that, what we need to do, we need to compare our disease group with the control group. Okay. We need to compare our disease group with the control group. So we need to compare the control group versus the disease group. So if there is any statistically significant difference is there, then we can say that disease got induced. Okay. So that is our primary objective. Then what is the secondary objective in our study? Whatever the test drug I have selected, whether that particular test drug can able to decrease the disease or not. That is my second objective. So that is, so for this objective, what I have to do, I have to compare my test data with the disease data. So hope you understood. So here 2 comparisons are very important. What is the first thing is, first objective is first of all whether disease got induced or not. So for that, what I have to do, disease data I need to compare with the test data. Another objective is whatever the test drug I have selected, that test drug is having, test drug can having the whether it is having any ability to attenuate the disease or not. So that's why for that, what I'm doing, test versus disease. Another one is the disease versus control. So see here, if you will select the none, it will not compare any group with another group. So for example, see, compare the mean of the each column with the mean of the every other column. So here normal group will compare with the disease, normal group with the standard, normal group with the test. Like that, it will give all the permutations and combinations. Otherwise, sometimes what it will, you may need, we will compare all the columns against only control. Okay, so we will here compare the mean of the each column with the mean of the control column. If we want to compare only with the control column, then go and opt this one. Okay, so for example, if you opt this one, so for example, like compare the mean of the each column with the mean of the control column, then what it will do, it will see what it will do, it will recommend the done net test. Okay, I hope everyone is aware about the post hoc test. Like here different tests are there. See, done net test, Bonferroni test, SIDAC test, like the different tests used there. So whether we need to select done net or Bonferroni, we have to choose. So if you will select this one, what software is suggesting, that done net is the recommended one if you want to compare our data with the control column. No, if I want to compare every column with the another column, then what it is saying, Q key is the recommended one. So here also, if you see, if you will select this column, then multiple tests are coming, Q key, Bonferroni, this Q key, Bonferroni will come. So Q key is added additionally. So if you want to compare all the column versus all the column, then it is recommended. Q key one is the recommended. So then if you will go for the, and also see one more important thing is the, so generally in all the data, we will select the 95% confidence interval. 95% confidence interval means 0.05 we will go. Okay, so we need to select the 0.05, 95% confidence interval. It is not compulsory that we need to go for the 95%. Based on your study, we can go for the 90% or we can go for the 99%. It is absolutely, it is on us. Okay, then just click the OK button. So it is performed the analysis. See, if you see, like this is the data table it will generate. So first I will, there are two data sets are there, ANOVA results, as one, another one is the multiple comparisons. So if you see this one, multiple comparison test, see, so here what is the control versus disease data, control versus standard data, control versus test. So control versus all the groups, then disease versus standard, disease versus test. So all the data sets for disease. So what is our first objective? Is there any significant difference between the control versus disease? Means first, so what is our objective? Whether disease got induced or not. So I have compared control versus disease. Is there any significance or not? Significant is there or not? What it is showing? What it is showing? Is there any significant or not? Is it showing yes or no? Any guess? Any answers? Control versus disease. Is there any significance? What it is saying? Yes or no? It is showing yes. So if it is yes, if it is showing yes, it will give the stars. Okay. So am I audible? Yeah, thank you. So first objective got fulfilled. Okay, so yes, there is a significant difference between the control and disease, means it is saying that disease got induced. What is the next objective? Whether my tested drug is acting or not. So for that, what I have done, so disease versus test comparison also I have done. So when I have compared the disease versus test, then also yes, it is statistically, it is showing statistically significant difference. So for that also it is showing the stars. So I have to keep these stars on my data table. Simply it is also generate the table graphs also. See, like here, like which kind of graphs we need to select, like based on the journals, like different journal. Of course, it is like not only based on the journal, based on the author also. So a few people, they want to, first I will go for the summary data. So see, so this kind of data set will. So if you go for the summary, like all the, like the mean plus or minus SEM data will. Okay. Hope it is visible. So graph will be developed like this. Okay, if you will go for the group data. So instead of the group data, for example, like so we can also go for the individual values. So if I will go for the individual values, it will not come mean. So whatever the data sets, for example, let us say that So graph will come like this. Okay. So each dot represents the individual value. Okay, each value represents the individual. So like based on our interest, we can go for the individual values or we can go for the grouped values. So this is about the statistics. So best available tool is the GraphPad. So like please, I think everyone is habituated to the GraphPad. So if you have any doubt, please contact me. I'm happy to share. I'm happy to help you. Okay. So then remaining things I don't want to go extensively. So in case of the behavioral data, we have done different experiments. So open field test first we have performed to explore the locomotor activity. So in this particular test, what we will do means like virtually we will divide into the system space. So how many numbers animal is like within, like we will set some cutoff time, maybe it is 5 minutes or 8 minutes, like that. Based on the experiment it will vary. So how many number of squares animal can able to crash within this particular 8 minutes we will measure here. Okay, so number of squares. So for example, if you are performing any anxiety related experiments, then how many number of central squares animal is crossing, how many number of peripheral squares animal is crossing. So this is about the open field test. So these are the trackings. So trackings also we will get the trackings by the total number of entries. So number of alterations we see, for example, like first animal enter into the this arm. Next, after that, animal entered into the this arm. See now, like if animal is entering into the this arm. So this is the alternative entry into the alternating arms. So A, B, C. So this is the number of alterations. So A, B, C we will give as a 1. For example, instead of that, if animal is first it is entered into the A, then maybe it will enter into the B. Again, if it will enter into the A, it is A, B, A. A, B, A means repetitions are there. A, B, C, it is not alteration. So we will not count that particular entry. So whenever animal is entering arms, then only we will consider it. So these are the trackings for the by image. Then already I have explained about the Morris water image. So here what we will see. So if animal is having memory, then within short span of time, it will reach the hidden platform. Okay, it will reach the hidden platform. This is called as doesn't have memory. Then it will take more time to reach the platform. So by using Morris water image, we have developed a number of entries, time spent in the target quadrant, then distance traveled by the animal. So all this particular data we can able to generate by the Morris water test. Then after completion of the behavioral test, we have sacrificed the animal. Then we went for the biochemical studies, molecular studies, as well as the histopathology. So as I said, like in case of the Alzheimer's, the levels of the neurotransmitter, especially acetylcholine levels, used to be very much reduced. So that's why we have estimated the levels of the acetylcholine in the hippocampus. So that's why we have conducted. So indirectly, we have estimated the levels of acetylcholine by calculating the acetylcholinesterase activity. So this is the neurotransmitter we have estimated. Then we have calculated as oxidative stress as well as the nitrative stress playing very important role in the development of the Alzheimer's disease. We have also measured the different antioxidant tests. So for that, we have selected like malonaldehyde, like lipid peroxidation test. We have performed nitric oxide test. We have performed by using the free-staging method, and also we have estimated SOD as well as the GSH. So these are the different tests and their statistics. So like this is the data of the SOD, this is the data of the GSH, MD, like we have molecular test. So for the molecular test, we have conducted the ELISA test. So here, like, what are the main, like, we went for the, like, sandwich. We have done the sandwich ELISA model. So this is the brief procedure, how we have conducted. So these are the main parameters we have estimated. As ELISA is very much costly, so we have, we have, as BDNF is the neurotrophic factor, so how it is expressing. So then I told you already, beta amyloid plaques. Okay, beta amyloid plaques also plays very important role in the development of the Alzheimer's disease. So that's why we have estimated the levels of the beta amyloid by using the ELISA. So if you see here, this is the disease group. In case of the disease group, see the thing. This is the control group data. This is the disease group data. So in case of the disease group data, the levels of the amyloid beta were significantly elevated. Okay, significantly elevated. Itself, it is suggesting important role in the development of or progression of the Alzheimer's disease. When we have treated with our test drugs, the levels of the beta amyloids are significantly like BDNF levels we have performed. This is it plays neuroprotective action. In case of the disease group, these levels were significantly reduced. In case of the treatment group, this neuroprotective BDNF levels were significantly increase. So as BDNF and beta amyloids are playing very important role, we have done protein expression of these studies. Then also we have performed for the neuroinflammatory mediators like TNF alpha and NF kappa beta. Then we went for the Western blotting to see the expression of the proteins. So I think you all are aware that different blottings are there, like Western blotting, Southern blotting, Southern blotting, Northern blotting. So all the things are coming. So we have performed the Western blotting to determine the protein expression, See, like this is like I don't want to discuss in detail about the procedures. So this is the data what we have generated after completion of the Western blotting. So here beta secretases. I told you like beta secretases plays important role in the development of the it will place important on whether amyloid precursor protein metabolism, whether it has to go to the non-amyloidogenic pathway or amyloidogenic pathway. If beta secretases are levels were expressed more, then what will happen? Amyloid precursor protein go and it will choose the amyloidogenic pathway and it will lead to the development of the amyloid beta plaques. So that's why we have measured the expression of the base protein. So see, if you see base proteins are highly upregulated in the disease group. And if you see the treatment group, base protein levels were significantly reduced. So to determine the protein expression, we have performed the Western blotting. So in case of the Western blotting, again, we have taken only important parameters, important proteins. One is the beta secretases and another one is the NF kappa beta inflammatory transcription factor. Another one is the BDNF. We have measured all these three protein expressions by using the Western blotting. So this is the data for the Western blotting. Then, as I told you, last one is the histological studies. For that, we studied the histopathology. So these are the histopathology described here. Red arrow indicates the multifocal apoptosis or atrophy of the neurons. So in the disease group, we have observed the apoptosis of the neurons. See, completely it is normal. There is no apoptosis, no inflammation, none. So this is the data at the 4x level. If you see at the 10x level, so same we have observed the X, but in case of the see here, if you see the intact hippocampus at the 10x, 10 into 10 means 100x. So this is the CA1 region, CA3 region, then Prakash. So we are doing this project together. So please let me know if you have any kind of queries. Please ask.
Subject change and problem.
Yeah, any queries, if you need any clarity on anything, I'm happy to explain. Yeah, sir. Am I audible?
Yes, sir. Yes, sir.
Okay. Please, if you have any doubts, related to anything, not only related to this study, related to research, you can ask me the questions. Okay. If you have any queries, I'm happy to read. So even for referencing, whenever you are writing paper or thesis, referencing also plays very crucial. Okay. So please use Mendeley, that is one of the best software for referencing. Okay, previously during our PhD days, we used it to EndNote. So EndNote is also good, but we need to have subscription. But Mendeley is the open access. You can go for the Mendeley, and also nowadays people are saying one more thing. It is Jutero, something like that. That is also good for the referencing. So you can use all these softwares for reference. So be thorough with all the softwares statistical softwares and referencing softwares. And also to draw the diagrams, you can use different software, you can use different softwares to draw the diagrams also. Okay.
Hello, sir.
Yeah, I think someone.
Contact factor and side score, sir.
Okay. Yeah. Site score and impact factor, it is like, see, what I can say, different agencies will give different statistics. Okay. So for example, like, as we are aware that Thomson Reuters, like, of course, Thomson Reuters now hand over by the Clarivate, they will measure the quality of the paper in impact factor. In case of the site score, Scopus, their measurement for the quality of the paper is the site score. So as per the Scopus, they will take the site score. As per the Scopus, they will give the site score. Web of Science, they will give the impact factor. So, but in our, what I can say, academic fraternity, people used to prefer both and preferably impact factor. Everyone used to ask, like, what is the impact factor of that paper, like that. But of course, two things are important: site score as well as the impact factor. Site score will be given by the Scopus. Impact factor will be given by the Clarivate, previously known as the Thomson Reuters. So this is the difference between the site score and the impact factor. So, like, for example, if I want, like, so is it clear, sir? Is it clear?
Okay, sir. Okay, clear, sir.
Yeah, thank you.
Good evening, sir. Good evening, sir. Hello.
Yeah, good evening.
Sir, any new drug molecules are there in clinical trials, sir?
Yeah, can you please, like, I'm not audible. Can you bit louder, please?
Any new drug molecules in clinical trials, sir, for the management of Alzheimer's disease?
Yes, this PPR pioglitazone, it is under the clinical trials.
Pioglitazone. Okay. Thiazolidine ion derivatives, no, sir?
Yes. PPR
Okay.
alpha derivatives
Rosiglitazone,
now, like,
pioglitazone.
yeah, I think pioglitazone, it was under the clinical trial. Maybe now few more drugs also maybe under the clinical trials. Under this category, like PPR alpha category, yes, few drugs are under the clinical trials also for Alzheimer's disease.
Okay, sir. Okay. Sir, what is the significance of IC50 values, sir? Did you calculate the IC50 values of the molecules, sir, in your study?
Yeah. Yes, exactly.
For
Like,
the potency
IC50,
only,
like, no, like IC, yes, of course, it is for the potential. Like, it used to vary based on the studies. Okay. So, for example, like, what I can say, especially IC50 values, it is important for the new chemical entities. That too, when we are going for the in vitro studies. In case of the in vitro studies, we will calculate the IC50 values. So, the concentration at which we are observing the 50% lethality to the cells. Okay. So, in case of whenever we are performing in vitro studies, we will calculate the IC50 at first. Okay, for that we will go
compared
for
with
the
the
MTT
standard
studies.
molecule. Okay.
Yeah, like, for that, like MTTSA, by using MTTSA, we will calculate the IC50 value.
Okay. Yeah,
When we are
anti-cancer
conducting
studies.
in vivo SS, when we are conducting in vivo SS, then LD50 is valid. LD50, lethal dose. So if
Sir,
it
one
is
more last one.
then we need, yeah, if it is new molecule, then we need to calculate the LD50 value by using OECD guidelines. OECD, Organization for Economic Cooperation and Development. By using OECD, we need to calculate the LD50. We need to identify the LD50 of that particular drug molecule. Based on the LD50, 1/5, 1/10, and 1/20th of the LD50, we need to select as a starting doses for our pharmacodynamic study. So IC50 for in vitro studies, LD50 for in vivo studies.
In vivo. Okay. And last one question, sir. For antioxidant screening, in vitro models are there, sir, from ABTS model, DPPH, and nitric oxide scavenging activity models, sir. And any other important models, sir? Other than 3, any other important models, sir?
No, DPPH is the standard. If you perform at least that one, that is sufficient. No need to go for more. Like, DPPH is the standard. That is more than sufficient.
Okay. Thank you, sir. Thank you for clarification.
Thank you so much. Yeah, please, like, if you have any kind of doubt, it is whether small or big, it doesn't matter. Like, I'm happy to clarify if I know. Yeah, very good evening. Oh, sir. Yes, sir. How are you, sir? Yeah, I'm good, sir. How are you, sir? Yes. Any doubts, sir? Thank you so much, sir. Yes, regarding like for statistics or for reference. Yes. Graph patterns. Yes. No, it is paid only, but unofficially we can get the crack version. Okay. Yeah, it is. No, free softwares, no, like SPSS is also subscribed one only, GraphPad also, Excel, like by using Excel you can calculate, but it is very tedious job. But easily you can get the crack versions if you want to use the GraphPad Prism. Yeah, thank you. Yeah, any more?
I think that's it, sir.
Yeah, thank you.
Thank you so much, sir, for gracing us with your knowledge and expertise. It's a very detailed description. Thank you so much, sir. And I also would like to thank all the participants for your active participation and thoughtful questions. Your engagement is what makes these FTPs so enriching. I would like to extend a heartfelt gratitude to the college management, our correspondent, sir, Mr. A. Raja Rammohan Rao Garu, and Principal Dr. A. Narendra Babu Garu, and the organizing team, my colleagues, for their continuous support in organizing such events and promoting educational excellence. As we conclude today's session, I want to express our sincere thanks to each and every one of you for your presence and active involvement. Your commitment to continuous learning and professional development is commendable. We look forward to having you with us in tomorrow's session also, where we will continue our journey for learning and growth. Until then, take care. Have a great day. Thank you, sir. Thank you so much, Pawan sir.
Yeah, thank you. Thank you so much for providing this opportunity. Thank you, sir. Thank you very much to Principal, sir. Thank you. Thank you to all.
Thank you, Pawan.